Our next speaker is Sofía Lavista Llanos. Sofía got her bachelor and PhD degrees in biology from the University of Buenos Aires in Argentina. After that, she joined to the Max Planck Institute for Chemical Ecology in Germany, where she has worked for the last 13 years, first as a postdoc and later as a project leader at the Department of Evolutionary Neuroethiology. And her group works on Drosophila neurogenetics, mainly studying the chemosensory specialization of various Drosophila species. I think the pronunciation is okay. And her talk is called Towards a Single-Cell Transcriptome Atlas of the Drosophila Olfactory Organs. So are you here, Sofía?
Great. Yeah. So good afternoon. This is the last talk, so please hold on there. It was a great talk just before, so thanks a lot, Patricia. And we will change completely topics here, and so I think you can relax. We will go back to biology. So I am a biologist, as you mentioned, and I've spent the last 12 years and a half, 13 years at the Max Planck Institute of Chemical Ecology, studying chemiosensation. That means olfaction and gustation in insects, basically. And so in the last year, we started with a PhD student that he's very eager into molecular biology to try to get some big data on this. And I will explain why I say big data. So today I will just show you the crude data, the raw data, which needs to be cleaned. And that's why I came here. And thanks for inviting me, especially Elizabeth Tapia. She gave a keynote talk yesterday, and she was the one that showed me this super congress to come and be part of it. So at the Department of Evolutionary Neuroethology, we mainly have two aspects of questions that we tackle, which is insect odor-guided behavior and the underlying neurobiological substrates. So that means smell, how the insects smell, and which are the neurons in the brain that direct that kind of behavior. And also the second one is the evolutionary aspect, how the olfactory functions change with the ecological context. So in both areas I've been working, and today I will try to fuse everything in a very short presentation as an introduction, and then go to this particular project that it's still starting. So my work was mainly done in Drosophila melanogaster, even if the department uses many other species. And Drosophila is a great model for, it's a genetic model organism. The genome was published in the year 2000, but it's also a very interesting insect in terms of the wild type behavior that they have, or she has. And Drosophila melanogaster has many close related species which have a bit of a diverse behavior in terms of ecology, which is very interesting. And we use the fact that we can compare those because we can translate or transpose some of the genetic tools that they have been developed in melanogaster to these other species. So I just mentioned that we studied olfaction in insects, and it's true, the insects smell a lot of compounds and they are really good at doing that, and they depend on this behavior, on this capacity for their survival. So here we see the head of a Drosophila melanogaster and the two olfactory organs which they use to detect these volatile chemicals or compounds are the antennae and the maxillary pulps, here depicted in green. So these organs are covered by hairy structures called sensilla, and we see here a zoom in these structures. Typically, melanogaster has 4 types that they are described by their shape. So we have basiconic, xyloconic, tricot intermediates in the antennae, and basiconic in the pulps. These hairy structures are actually very hard in cuticle, but they have diminute pores where the molecules will come in and make contact to the dendrites of the olfactory sensory neurons that are housed here inside. And then they will contact the olfactory receptors. So the vertebrate olfactory receptors were described in 1991, and Richard Axel and Linda Buck, and they got a Nobel Prize actually in 2004 because of this discovery, but it took 10 years for the insect olfactory receptors to show up. And that's mainly because they are quite different or divergent. Even the topology of the proteins is completely opposite. So what happened is that in the year 2000, I said the genome was released, and almost 1 year or some months before, by mining the genome database and looking for a type of family of proteins, 3 groups in parallel described actually, or came up to the family of these divergent 7 transmembrane domain proteins, which were candidate to be the odorant receptors in Drosophila. And one of them actually by in situ hybridization showed that indeed they are expressed in the neurons that are housed in the antennae and maxillary bulbs. Very early work at the department done by Dieter Wischer here. And also there is a second group of Leslie Boschel at the Rockefeller University in the United States. Both of them, they published a Nature paper where they described was coming about. So there's olfactory receptors are actually formed by dimers. So we have a very conserved monomer Orco, or olfactory receptor co-receptor, that is always being expressed with a second monomer, which is highly divergent, and it's the one that gives chemical sensitivity to the complex. I will not go into the details, but there is much more information and research that Dieter and others we have been done in terms of the signaling cascade that goes on after the odorant. In this case, ethyl butyrate, for example, the compound is being or eliciting an olfactory response in the OR22A receptor. So these ORs have been very well characterized. There is a database, DOR database, that one can look. And for example, here I just made a heat map as an example where we have many of these ORs, many of the 60 or 62 proteins, 60 Drosophila, and different compounds. And we see that there are some ORs that get actually inhibited by many of the compounds. Other ones get very activated by those, and not a single OR is going to be elicited by every compound, and not every compound will stimulate every single OR. So types, and both 2 extremes would be very generalist ORs, where we see that many ligands would activate these complex receptors, or very specialized, like the case of geosmin, very interesting case also, where only one compound, one single compound is almost like a pheromone receptor, is going to give a response. So Veit, he's a postdoc at the lab, and from the group of Silke Sachse of coding, and together with him, also there is a description today of how is that all these different olfactory receptors are being expressed in each of the types of the sensilla and olfactory sensory neurons. So each olfactory sensory neuron will express one or a few of these ORs, and they have the shape that we just described. They express, for example, intermediate pheromone receptors, basic onyx ORs, and they are a little bit linked with the categories of compounds they can detect, or they do detect. And once the chemical binds to the receptor, there is a chemical signal translated into electrical signal of the olfactory sensory neurons, and this information gets translated or transposed to the brain. Here is the brain of Drosophila, and so we have different lobes, the antennal lobes, the glomeruli, and different structures in the brain that will receive this information, and via projection neurons and local interneurons, bring it to other higher centers, the mushroom bodies for association, and the lateral horn for codification. And this is the work done by Silke Sachse. And so she has an independent group, but together with the department. And so there in the brain, there is this odor code where a compound or a group of compounds, a bouquet of different compounds, will get a category of good or bad. And that actually will depend a little bit also on the internal state of the fly. So I'm going very fast here. In the lab, we talk about the valence of an odor. So it's going to be an odor that elicits aversion. For example, this geosmin compound that I mentioned before is produced by mold. Geosmin is not toxic per se, but the mold is really toxic for the flies. So it's going to be alerting the flies not to go there. The same happens with these predator wasps, that the fly uses some of the compounds that the wasp has or produces to try to avoid oviposing on these areas. And the opposite is going to be an attractive odorant like food or yeast, where flies really like to feed and lay eggs, or even pheromones for mating, finding matings. So my work was also much into the ecology and how the chemosensory aspect can really shape the niche of a fly or of a species. I will not go into detail, but here we have Drosophila cercellia. Is a very close species to Melanogaster, and she is a very highly specialist in the fruit of noni fruit or Morinda citrifolia fruit. So this fly finds the compound here, methylhexanoate, very much attractive and virtually ignores the two acids here, which are hexanoic and octanoic acids, which, by the way, they are very highly repellent and toxic to virtually all the other specialists. So two compounds here are separating exactly the niche of these Drosophila cercellia that it's going to be thriving in Morinda citrifolia, and the other ones that are going to be dead there. So in spite of all this research today, we still don't have an olfactory code. And by that, I mean the link between the structure of an odorant, an odorant structure, the neural activity, and the odor perception. We cannot, but just by looking at a molecule, we cannot predict how it will smell. This is also a question that was assessed in a very big project that just finished last year, the nanosmell project. So this PhD student wanted to go back to basics, and this is defining here the olfactory unit. So we talked about the olfactory receptor neurons, which are these gray neurons here, but actually these neurons are sitting on a kind of a syncytium unit, which together with many other type of cells. So he said, maybe we have to go back and crack in here. So far we know quite a few genes that are being expressed, like the olfactory receptors, odorant binding proteins, and many other ones, but still they are very few. So what he wanted to do, and what he's doing, is to take the antennae of the fly and try to make a characterization at the cellular resolution of the profile of expression by RNA sequencing. So what he did, here's Sinisa Prelik, the student that he's doing that. He collected many, many antennae, and then he cracked them open, a bit of mild digestion with papain, and then he extracted the cells and collected them on a tube. And then we decided to go for a single cell approach using the chromium controller of the 10 times genomics. This is a technique that it's not so new anymore. So, but still it's quite expensive to use. So at the Max Planck, we got funding to do it, but it's not something that it's on regular use at the moment. And we did it actually at the Genome Center, at the sequencing center of FLEI Institute, that it's there at the same campus of the Max Planck Institute with Marco Groth, he's the director of this facility. So we just have to bring the sample and pipette it here in the sample well, where it will get combined with a gel well. Now I'll show you in a minute. And then here, everything is recovered and sequencing happens like magic. So we have droplet encapsulation. So the idea is that we will have oil droplets that keep coming, and then the aim is to get one bead collected together with a nucleus or a cell, a single nucleus or single cell. And once this happens, all the enzymes and reactives necessary for the barcoding of the mRNA and for the cDNA, for the retrotranscription and for the library preparation and sequencing, everything is included here. And each bead will get a cell barcode. So, and then we will have these unique molecule identifiers. So at the end, after the cDNA synthesis, sequencing, and alignment to the genome of Drosophila, then the cell ranger, which is everything mostly done by the facility, will collect all the molecules that belong to each of the beads, and then they will call them different cells or different nuclei, depending on the sample that we are trying, and then create a matrix of expression. So this is the ideal situation where we get, or the system tries to find from all the beads that they were sequenced, many of them are going to be empty or empty droplets of oil. So we get a collection of cells that are real cells, and then we get another one that are backgrounds. And we will work with this top here. This is how it should look. And this is our first sample. We made one biological sample, two technical replicates so of antennae of male Drosophila, male antennae. And what we see is that actually we got a very few number of cells difficult. We were expecting much higher cells. My connection says it's unstable, so I hope you are still with me. So we have the second case,
yes, we are
and we
here.
have the situation. Yeah, perfect. I get some messages that it's a bit unstable. So there are many reasons. I got very disappointed when we saw the data, but it seems that it's more common than what one would expect to get this type of data. We have to remember, if you know, Drosophila, I didn't mention, is the small fly that is flying next to your bananas in the kitchen. That's the fly for somebody that doesn't know it. And from there, we get the antennae, and from those, we have to get single cells. So it's an extremely complex structure that we are working with. Anyway, so once all the data is collected, we continued, even though the data doesn't, it seems that it's going to be hard to get and to clean, as we just heard, to clean out the noise. But anyway, so after a principal component analysis, the data is shown in a two-dimensional plot, and we have a first clustering of this. And so what Sean did was to go quickly and look at the identifiers genes. So we have information about the antennae. We know that they are neurons. We know that they are epithelial cells. There is a muscle, the antennae moves. There is hemolymph and support cells. And there is also hearing and mechanosensation going on in the antennae. So we know what to expect a bit, so that we can use to try to ponder how good or bad our data is. And after that first subclustering, we got 1,351 transcriptomes that they are clustered in 9 clusters. And so going through the clusters, CNISA could actually, not guess, but could identify some of these clusters. They would feed to muscle cells, for example, and another ones to epithelial cells. But we have these other situations in which we have one cluster that we cannot say much about it, because we don't see genes that are very highly represented. There it's like a medium of everything. Then we have these other 2, which we can find genes higher or lower, but per se, we don't have a pre-identification of which type of cells they would be. And that is actually good news, because one of the points of this project is to try to make an unbiased identification of the cells. So far, all the olfactory field has always centered everything on an OR. So basically the neuron, the olfactory neuron, is just a neuron expressing an olfactory receptor there, and that's it. And we want to explain the olfactory code with just that, with one OR channels, but we don't have information about much information that we need to try to understand what's going on really. So yeah, at the beginning I thought it was disappointing. I find it that it's good to get that type of information. So, but anyway, we can go and look for those genes or transcripts that we know that they should appear, and have a look in the clustering, how do they show up. This that it's expressed in the olfactory sensory neurons that express ORs. We had the coeloconic ones I mentioned, they express another type of receptors, which are not co-expressed with Orco. And we see indeed that we showed Orco is coming up in basically in cluster 2, a little bit in cluster 3 and cluster 1. So if we keep looking at these ones, now we can see other genes that we would expect or not to be there, and try to see how they behave. So LAV, it's a neuronal marker. So if Orco is being expressed in the olfactory sensory neurons, we should expect to have LAV, and this is the case mainly in cluster 2, but also in cluster 3. Fruitless is a marker for the neurons that are being involved in the response to pheromone. And so we also find them, and there are very few neurons. So actually we find it here as well. Synaptobrevin is a synaptic marker also belonging to the neurons. We find it there. And then if we go to a transcription factor that it's mainly expressed on coeloconic ones, the ones that I just said, they were not OR expressing, but IR expressing. And we see that it's virtually at here. And IR8A and 25A, which are common ionotropic receptors, not from the olfactory receptor family. Actually, they show a little bit here in cluster 2, both of them. So it could be that cluster 2 is actually involving all the olfactory sensory neurons, the ones that express ORs and the ones that express IRs. And what's nice there is that if they are clustering, they are not doing so, but the OR and IR, because those we know are different. So I would like to know which are the transcripts that are making this cluster, that are putting all these cells together. And that might be an identifier of odorant neuron, that it comes higher from the OI and IR hierarchy. So to the data and the low fraction reads that we got. So of course, they provide us this facility and Marco growth here, very good. And so we try to find some explanation about it. And the main problem in this type of studies, where one is crushing all the tissue, is ambient RNA. Of course, cells are being broken up and RNA is floating around. And then what happens is that if we go back to our little bubbles or drops of oil, we thought, or in theory, we have a bead and a very clean nucleus, but actually we have ambient RNA going around, and it will get here inside and kind of contaminate or give background to our samples. And this is probably what's happening with us. So now we can go, and instead of looking to the cells, we can have a look to the transcripts. And so maybe we could just select those genes that are high represented in this unique molecule identifier. That means that they are high in the in quantity in the sample per se, and look for those that they have barcodes, so they have been associated both to empty droplets, the ones that don't have the nucleus, and the ones that the non-empty droplets. And so if we have genes that they have more or less the same kind of representation in both empty and non-empty, we can speculate that they come from the ambient or check how they behave. Here we have the 9 clusters again. OBP19 is one of these odorant binding proteins, very highly expressed in the antennae, and we see that actually it's present ever. The same happens with antennal 10, which is also a highly expressed gene, and with OBP83B. So actually, it's not that these genes, probably some of the clustering here, it's true, and they belong to this cluster, but they are also contaminating. It's not informative at the moment, so we can ignore them, let's say. And so we do the opposite here. We look for those that they are droplets. And so we got some, nothing I would say canonical of olfaction. And so and they were very few. So what we did now, it's okay. We have sample. It seems that it didn't so well because we have some ambient RNA. So what about if we comparing to bulk RNA? There are many studies that where they just crushed out the intent, caring of which cell the RNA comes from. And when we do that, we see that actually the studies compare our data with from the 15,000 genes that they are in Drosophila, they get like 8,000 or 9,000 genes being expressed in the antennae. And from that, we get 1/3. So it's either that our sample had fewer cells that we speculated, even though we counted them, or that everything got a bit crushed. And so this ambient RNA is kind of masking our transcripts.
Sorry, Sofia, we have 2 minutes left.
Yeah. Okay, so I'm almost done. So yes, the second aspect that can happen is that the variability between the cells can be higher than the one that the cell ranger is kind of countabilizing, and one can bypass that and asking the system to force the call and say, okay, give me 10,000 cells and see what happens. So that's what we did, and now Sin is analyzing this data. We just got this plot, and so we have a clustering here. We have 11 clusters with that and with data, but this is not going to take out the ambient RNA. So this is my last slide. This is two years. We are working towards a single cell transcriptome class in the antennae. And I said that I was in Germany. So last year, from January this year, very happy that I'm a guest scientist at bioinformatics group of Elizabeth Tapa, the CIFASIS in Rosario, in lockdown in Buenos Aires. So we work by Zoom and like this conference, but have other projects going on together with her, and they are working very well. Now we can boost a bit this project and help Sinisa that he's back in Germany. And here I show you just one example of an immuno, which is in green, and then NOCH is a gene that is characteristic of one of these supporting cells. So this is what we are aiming for. We want to do this with all the genes, especially the novel genes that we will discover being expressed either in the syncytium, in the olfactory unit. And with that, I just finished thanking the Max Planck Society for the funding, the Department of Evolution and Neuroethology, Bill Hansen, which is the director, Sinisa is here, the PhD students, Marco Grott from the FLI facility, and Sofía here at CIFASIS. Thank you.
Thank you so much, Sofía, for your nice presentation. I have learned a lot about Drosophila. I never worked before with these organisms. And so I was ignoring a lot of information that you teach us today. So thank you so much. I will check if there is some question.
Yeah.
There is no question at the moment. I have a small question because I have been, well, there is a question. Yes. I will prefer to do the question from the audience and then my question. So Raquel Alvarez says that RNAs are so sensitive to degradation. So how do you know that you are capturing the transcription of those individual cells in response of the odor and not as response of cell damage? So the process of isolation the cells for performing the single cell analysis.
Yes, sure. Yeah, it's very highly sensitive. And actually, I missed it. So like the RNA that it's being exposed to an odor. We're just taking flies that are grown in normal medium on a tube via where we grow them and we collect. Not looking for we are not comparing here genes that are coming up or down depending on the, just want to know the basics, because so far we have a lot of information about olfaction, but at the molecular level, the expression of genes, there is of course many papers being described, but it's basically the ORs, they'll kind of modulate the olfactory response. And we would like to try to tackle everything of that. And of course, we used a system we can go and do in parallel RT-PCR or quantitative PCR, look for genes that we know that are being expressed in 10E, and as a control, we have like detect Orco or OR22A, and we check that the sample is not being degraded on the way. Of course, there are other, and you can keep them there, wash them, and then rehydrate them. But the point is that at the moment before pipetting, the cells can be lysed, and it's not a unique problem of us. It's a problem that it's very highly contemplated in the technique, in the technology, and I'm attending some Zooms where they are trying to, circumvent this.
Oh, thank you,
so
Sofía.
It looks magic, but in practical, it's, we have to be careful.
Yes, thank you. We have another question from Vanina Villanova that is saying, why do you use nuclei and not complete cells?
Because if you remember when I showed this cartoon of how this works, and at the beginning of how the olfactory system, we have the olfactory sensory neuron that it's being actually sitting under the cuticle. The dendrites are coming up to the shaft, and then on the axon is going all the way to the brain. The moment I cut the antennae, the olfactory sensory neuron, it's gone as a cell. So this is a tissue, and it's very, very complex tissue. So it's easier to collect nuclei than to collect cells. And I was in cell and nuclei in the slides, and I did that because it's actually, we call them entities. Yeah. So in our case, we're using nuclei intact cells, particularly the olfactory sensory neurons. So we are cutting them. There is actually Dieter, they have a protocol, they came after many years where they can kind of not grow them, but keep them a little bit on. One of the ideas to try to collect them there, keep them on a plate, and then try to use complete cells. That would be great, but it's very hard to get them.
Yeah, sure. Thank you. We don't have any more questions. I will do my own question because I never worked before with single cell RNA-seq. So my question is about the replicates that you obtain. Where do you combine the information from the replicates before doing the PCA or?
After. In this case, I did it after, but in this case, I'm sorry, this is the very first run, and I decided to show the data, even though it's very raw. It's one biological sample, which actually the biological sample is 110. So it's 110, so we could say N of 100 if you want, but pulled together, and 2 technical replicates. And the ones that I was pulling was at the end, after all the process. So we treated them as independent samples, and then at the end we combined these transcriptomes together.
And you will have different barcodes for each cell from each sample, really?
Yeah, yeah.
Okay.
Yeah, because they were pipetting parallel. Yeah, that's true.
Okay, perfect. So we don't have any more questions. Thank you so much for your presentation. Thank you to all of you that are attending this session. And thank you for inviting me.
Thank you.